Hello everyone and welcome to our CUET PG 2026 preparation lecture on Plant Biotechnology. In this session, we are going to study Unit 3 which focuses on Protoplast technology, Somatic hybridization, Cybrids and Somaclonal variation. These topics form the foundation of cellular plant biotechnology and are extremely important from an exam perspective because they explain how plant cells can be manipulated directly under laboratory conditions. Traditionally, plant improvement depends on sexual reproduction and breeding, but modern biotechnology allows scientists to combine traits, transfer genes, and generate variations at the cellular level itself. This unit will help you understand how cell walls are removed, how cells are fused, how hybrid plants produce, and how genetic variation arises during tissue culture. These are not just theoretical ideas, they are real techniques used in agriculture and crop improvement. To understand protoplast technology, we must first understand what a protoplast actually is. A protoplast is a plant cell from which the cell wall has been completely removed. In normal plant cells, the cell wall provides mechanical support, protection, and structural rigidity. However, this rigid structure also prevents direct manipulation of the cell, especially cell fusion and DNA uptake. When the cell wall is removed using specific enzymes, the remaining structure is a living cell enclosed only by the plasma membrane. This naked cell is highly flexible and can interact directly with its environment. Because of this property, protoplasts can fuse with other cells, accepting foreign DNA and regenerate into complete plants. This is why protoplasts are considered powerful tools in plant genetic engineering and cellular manipulation. Protoplasts have several unique characteristics that distinguish them from normal plant cells. Since they lack a rigid cell wall, they are surrounded only by the plasma membrane. This makes them extremely sensitive to osmotic conditions. Even small change in surrounding solution concentration can cause swelling, shrinking, or bursting of the cell. Therefore, maintaining osmotic balance is crucial and critical during handling and culture. Another important characteristic is their regenerative capacity. Even though the cell wall is removed, protoplasts are capable of synthesizing a new wall and resuming normal cellular functions. Under suitable culture conditions, they divide, form cell clusters, and eventually regenerate into whole plants. This ability reflects the principle of totipotency, meaning every plant cell has the complete genetic potential to develop into a whole organism. Protoplast technology plays a central role in modern plant biotechnology because it allows direct access to the living plant cell without the barrier of the cell wall. This makes it possible to perform somatic hybridization, where cells from different species are fused to combine their genetic material. This is particularly useful when two plants cannot be crossed through normal sexual reproduction. In addition, protoplasts are used for gene transfer experiments, membrane transport studies, and cytoplasmic inheritance analysis. Scientists can introduce foreign DNA directly into protoplasts and study gene expression. Protoplast technology has therefore become an essential tool for crop improvement, genetic engineering, and fundamental research in plant cell biology. Protoplast isolation is the process of removing the cell wall from plant cells while maintaining the viability and structural integrity of the cell. The aim is not just to dissolve the wall, but to obtain living cells that are capable for metabolic activity, division, and regeneration into plants. This requires careful control of enzymatic treatment and environmental conditions. The success of many downstream applications depend on the quality of isolated protoplast. If the plasma membrane is damaged or osmotic balance is not maintained, the cell may die. Therefore, protoplast isolation must be performed under sterile conditions with appropriate osmotic stabilizers and controlled enzyme exposure. Two main approaches are used to isolate protoplast: mechanical and enzymatic method. Mechanical isolation involves physical removing the cell wall by cutting or squeezing, whereas enzymatic isolation involves digestion of cell wall components using specific enzymes. Among these, enzymatic isolation is the preferred method because it is more efficient and produces large numbers of viable cells. Mechanical methods are difficult to perform and often damage the cell membrane. Therefore, the modern plant biotechnology primarily relies on enzymatic digestion for protoplast isolation. In the mechanical method, plant tissues are first plasmolysed by placing them in a hypertonic solution. This causes the plasma membrane to detach from the cell wall. The wall is then carefully cut using microtools, and the protoplast is gently released. However, this method has majorly limitations. It is technically demanding, produces very few cells, and frequently damages the membrane. Because of low efficiency and high risk of cell death, mechanical isolation is rarely used in modern research laboratory. The enzymatic method uses cell wall degradating enzymes to dissolve structural components of the wall. Cellulase breaks cellulose fibers. Pectinase dissolve the middle lamellae that binds cells together, and hemicellulase breaks hemicellulose networks. As these components are digested, the wall gradually weakens and eventually dissolves completely. Intact protoplasts are released into the surrounding medium. Because of the process is gently and controlled, most cells remain viable, making the method high efficiency. After enzymatic digestion, the solution contains intact protoplast along with cell debris and broken cells. Therefore, purification is necessary to obtain healthy cells suitable for culture. Filtration removes large debris while centrifugation separates cells based on density. Density gradient centrifugation allows selection of only viable protoplast. This ensures successful culture and regeneration. Several biological and physical factors influences isolation efficiency. Different plant species vary in cell wall composition, affecting enzyme action. Younger tissues are preferred because their walls are thinner and easier to digest. Environmental factors such as enzyme concentration, temperature, and pH also plays important roles. Osmotic stabilizers must be used to prevent membrane rupture. Proper optimization of these factors is essential for successful isolation. Once protoplasts are successfully isolated and purified, the next step is culturing them under controlled laboratory conditions. Protoplasts cannot survive in ordinary water or nutrient solutions because they lack the protective cell wall. Therefore, they need a cultured in specially designed nutrient media containing carbon source like sucrose, vitamins, amino acids, and plant growth regulators such as auxins and cytokinins. These components support cellular metabolism and stimulate division. Equally important is the presence of osmotic stabilizers such as mannitol or sorbitol in the culture medium. These components maintain osmotic balance and prevent the flagellate protoplast from bursting. Under optimal conditions, the protoplast begin to synthesize a new cell wall, regain structural stability, and prepares for cell division. Thus, proper culture conditions are essential for transforming an isolated cell into a regenerated plant system. After culturing begins, Protoplasts undergo a series of developmental stages that eventually lead to plant regeneration. The first stage is cell wall regeneration. The cell synthesizes cellulose and other wall components and forms a protective layer around itself. Once structural integrity is restored, the cell becomes capable of mitotic division. Repeated cell divisions produce a small cluster of cells called a microcolony. These microcolonies grow further and develop into callus tissue, which is an undifferentiated mass of dividing cells. By altering hormonal balance in the culture medium, callus cells differentiate into shoots and roots throughout organogenesis or somatic embryogenesis. This process ultimately produces a complete plant, demonstrating successful regeneration from a single protoplast. Totipotency is one of the most fundamental concepts in plant biotechnology. It refers to the ability of a single plant cell to develop into a complete plant under suitable environmental and nutritional conditions. This means every living plant cells contain the entire genetic information required for formation of all tissues and organs. This property is the biological basis of tissue culture, micropropagation, and protoplast regeneration. Without totipotency, isolated cells would remain isolated and could not develop into organism. Totipotency reflects the remarkable plasticity of plant cells, allowing them to de-differentiate, divide, and re-differentiate into complex structures when provided with the correct hormonal and environmental signals. Somatic hybridization is a technique in which protoplasts from two different plant species or varieties are fused to form a single hybrid cell containing genetic material from both parents. Unlike conventional hybridization, this process does not involve gametes, pollination, or fertilization. Instead, fusion occurs at the somatic cell level. Because this method bypasses sexual reproduction, it allows combination of genomics from plants that are otherwise reproductively incapable. The fused cells contain nuclear and cytoplasmic components from both parents. If successfully cultured and regenerated, it produces a hybrid plants that express traits from both parental lines. The primary objective of somatic hybridization is to combine desirable traits from different plants into a single organism. For example, one plant may have high yield but low disease resistance, while another may be disease resistance low yielding. By fusing them protoplasts, scientists attempt to combine both beneficial characteristics. Another important objective is the transfer of stress tolerance traits such as rough resistance, salinity tolerance, or temperature adaptability. Somatic hybridization can also be used to produce entirely new species, broaden genetic diversity, and overcome reproductive barriers that limits conventional breeding. Somatic hybridization involves several careful controlled steps. First, protoplasts are isolated from both parental plants using enzymatic digestion. These isolated cells are then brought in together under controlled condition that promotes membrane fusion. After fusion, a mixture of parental and hybrid cell is produced. Therefore, hybrid cells must identified as selected using appropriate markers or selective media. Once confirmed, hybrid cells are cultured and regenerated into complete plants through tissue culture techniques. Protoplast fusion can occur naturally when cells come into close contact, but spontaneous fusion is rare and inefficient. Therefore, artificial methods are commonly used to induce fusion. One widely used chemical method involved is polyethylene glycol (PEG), which alters membrane properties and causes adjacent protoplasts to adhere and fuse. Another method is electrofusion, in which an electric field is applied to align protoplast, and induce temporary membrane breakdown. When membranes reseal, the cytoplasmic content merge, forming a hybrid cell. Electrofusion provides greater control and higher fusion efficiency compared to chemical method. After fusion treatment, the culture contains many cells, but not all of them are truly hybrids. Some may remain unfused or partially fused. Therefore, identification of genuine hybrid cells is essential before regeneration. Several techniques are used for this purpose. Morphological differences may indicate hybrid formation, but more precise method includes fluorescent labeling of parenchymal cells, chromosome counting, and modern markers analysis. These methods confirm the presence of genetic material from both parents within a single cell. Once hybrid cells are identified, they must be selectively grown while eliminating parenchymal cells. This is achieved using selective cultured media that only hybrid cells can survive in. For example, if each parent lacks a different metabolic function, only few cells possessing both functions will grow. Antibiotic resistant markers are also commonly used. Only cells containing genes from both parenchymal survey under selective condition. This step ensures that the regenerated plant origins form true hybrid cells rather than parenchymal contaminants. Hybrids or cytoplasmic hybrids are special type of hybrid cells in which the nuclear genome originates from one parent, but cytoplasmic organelles such as mitochondria and chloroplast come from both parents. In other words, nuclear inheritance is uniparental, while cytoplasmic inheritance is biparental. This type of hybridization allows transfer of cytoplasmic traits without mixing nuclear genomes. Since many important characteristics such as cytoplasmic male sterility are controlled by mitochondrial genes, hybrids plays an important role in plant breeding programs. Hybrids are produced by fusing a normal protoplast with an enucleated protoplast, a cell from which the nucleus has been removed. Alternatively, after fusion, one of the nuclei may be selectively eliminated or lost during cell division. The resulting cell contains cytoplasm from both parents but retains only one nucleus. This creates a cytoplasmic hybrid that expresses nuclear traits from one parent and cytoplasmic traits from both parents. Such selective inheritance makes hybrids a useful tool for studying organelle genetics. One of the most important applications of hybrids is the transfer of cytoplasmic male sterility, which is widely used in hybrid seed production. Male sterile plants cannot produce viable pollens, ensuring controlled cross-pollination. Hybrids are also used to transfer herbicide resistance and disease resistance traits that are encoded in organelle genomics. Additionally, they help researchers understand mitochondrial and chloroplast inherited patterns and interactions with nuclear genes. Somatic hybridization has enormous potential in plant improvement. It allows genetic combination between species that cannot reproduce sexually due to incompatibility barriers. This greatly expands the genetic pool available for breeding. It also enables rapid transfer of desirable traits without multiple generation of crossing. Novel genetic combinations can be created that may never occur naturally. This makes somatic hybridization a powerful tool for developing improved crop varieties. Despite its advantages, somatic hybridization has several limitations. Regeneration of hybrid plants is often difficult because fused genomes may be incapable. Chromosomal imbalance and abnormal gene expressions can prevent normal development. Another limitation is low efficiency of fusion and regeneration. The process requires sophisticated laboratory techniques and skilled handling. Genetic instability in hybrids can also limit their practical agriculture use. Somaclonal variation refers to genetic variation observed among plants regenerated from tissue culture. Even when cells originate from the same parent plant, regenerated plants may show different in morphology, physiology, or genetic makeup. These variations arise because tissue culture conditions can induce genetic changes, while sometimes undesirable, these variations can also provide useful new traits for plant breeding and improvement. Somoclonal variation arises due to several molecular mechanisms. Gene mutation may occur during cell division in culture. Chromosomal rearrangements such as deletion, duplication, or translocations can also take place. Epigenetic changes like DNA methylation and activation of transposable elements further contribute to variations. Stress conditions in tissue culture, including hormonal imbalance and artificial environment, often triggers these genetic alterations. Detection of somoclonal variation involves comparing regenerated plants with the original parent. Morphological observations may reveal visible differences such as parent height, leaf shape, or color. More precise detection method includes cytological analysis of chromosomes and molecular marker techniques such as PCR-based markers. These methods identify genetic changes at chromosomal or DNA level. Somoclonal variation can be beneficial when it produces useful traits. Breeders can select variants showing improved disease resistance, stress tolerance, or yield. It also increases genetic diversity, which is essential for crop improvement programs. Tissue culture thus becomes a source of new variation of plant breeding. However, somoclonal variation is unpredictable. Many variations are undesirable and may reduce plant fitness or productivity. Genetic instability makes it difficult to maintain uniform plant populations. Therefore, careful screening and selection are required before using somoclonal variation in agriculture. In this unit, we have learned how removal of the cell wall produces protoplast capable of fusion and genetic manipulation. We studied somatic hybridization and hybrid formation as method of combining genetic material from different plants. We also explored somoclonal variation as a source of genetic diversity in tissue culture. Together, these technologies form the core of modern plant biotechnology and plays a major role in crop improvement and genetic research. To prepare well for CUET PG Plant Biotechnology paper, revise concepts regularly and practice questions for each unit. Solving MCQ tests and previous year papers helps you understand the exam pattern and improves confidence and time management. Biotechnology concepts become clear with regular revision and practice. Stay consistent with learning, revise often, and keep practicing questions to strengthen your understanding and perform well in the exam. Subscribe BioBrainzz for more.